Hi, my name is Daniela Sanchez, and I worked on the development of NEC4 biosensors for monitoring cellular activity this past summer. Some important vocab to know going into this experiment is that a kinase is a type of enzyme that adds chemicals called phosphates to other molecules such as sugars or proteins, and the process of reverse phase HPLC involves binding an organic molecule to a stationary phase in a relatively polar environment and then eluting the organic molecule using a gradient of a less polar organic solvent. NEC4 is a serine or a threonine kinase which has been implicated in cilia stabilization, DNA damage response, autophagy, and epithelial to mesenchymal transition. And these associations can demonstrate the potential of NEC4 as a cancer therapeutic target. The research aimed to investigate the interactions and specific functions of NEC4 using novel peptide biosensors, in which a total of 16 amino acid peptide biosensors were designed to selectively interact with the ATP phosphorylated active sites of NEC4, in which the effectiveness of the biosensors would be observed using fluorescent sensors in conjunction with enzyme assays. The hypothesis for this experiment would be that by elucidating NEC4's role in cellular processes, it would be able to be utilized as a therapeutic target against cancer. The FMOC protected species, including cyberesins, were deprotected in 30% DMF solution using a plate shaker at room temperature for around 15 to 45 minutes, in which samples were then washed twice with DMF, isopropanol, and DCM before performing a 9-hydrogen test to check for amines. Positive results would then lead to 3 equivalents each of coupling amino acid and oxyampure, which were dissolved in DMF. And after washing of the peptide was complete, a negative test result would indicate that it was completely coupled. After coupling 4-chloral-7-nitrobenzofurazan, the expectation was that the corresponding peptide would bond to it after overnight shaking, with TFA being the final solution added to protect the amino acids. Finally, HPLC liquid chromatography purification was used and characterized by mass spectrometry. The pi-pi stacking seen here between the nitrobenzofurazan group and the phenylalanine, common to all designed biosensors, disables fluorescence until phosphorylation of the intermediary threonine amino acid interrupts the interaction. The structures seen in the box toward the right represent all target biosensor sequences with either the NBD or danzo group attached. In these two graphs provided from HPLC, the top one shows NBDCL, which alone saw a peak at around 32 minutes, in which the same peak can be found in the bottom graph with all NBD peptides dissolved in methanol, indicating unreacted NBDCL. The peak collected at around 24 minutes proved to be desired peptide as later characterized by mass spectrometry, and the peak at 38 minutes in the surrounding area were more likely to be contaminants or byproducts from the injection. Mass spectrometry of the peptide collected in Figure 3 can show peaks at around 728 and 729, which closely resemble the calculated molecular weight of 726.79, which confirms correct synthesizing of the compound. Other peaks likely surfaced as a result of lack of concentration used for the injection solution, as this was a recurring issue during measurement. In this scheme, we can see that the deprotection of FMOC via E1CB elimination begins with piperidine attacking the tertiary hydrogen, in which the resulting carbon ion forms a neighboring double bond, promoting a series of electron pair movements producing dibenzofulvene, carbon dioxide, piperidine reformation, and the deprotected peptide. Coupling begins with carbamide activation with next amino acid and depronating the hydroxyl group and forming an OC bond. Subsequent substitutions with oxymopure and the existing peptide involve nucleophilic attacks on the carbonyl group of the FMOC protected amino acid. So, of the 6 biosensor peptides that were completely synthesized to completion and characterized by mass spectrometry, only one resulted in a final weight suitable for enzyme analysis, which would be 5.4 milligrams. However, this was still an extremely low yield in terms of the solid phase peptide synthesis. It's expected that the yield was so low because of the TFA cleavage cocktail, in which further synthesis could feature a different resin that's more compatible with the 95% TFA cocktail. Regardless of any prior interactions, if the peptide was not cleaved from the solid support resin bead, it would not be collected upon transformation from solid to liquid. And since different resins work best with different concentrations of TFA cleavage cocktails, future synthesis could feature rink resins, which are known to be more compatible with a 95% TFA cocktail. From the HPLC data provided, it's shown that between 20 to 30 minutes, the biosensor peptides tended to display its peaks, and characterization with mass spectrometry afterwards sometimes produced non-essential peaks, but were likely the results of injecting concentrations that were too low, as seen in Figure 3, and reruns likely could produce cleaner graphs. It's also curious that the HPLC graphs indicated NBD-Cl contamination even after washing between NBD addition and TFA cleavage. And given how much NBD stains, insufficient washing could also be another factor in the inefficiency of TFA cleavage, as any leftover NBD-Cl molecules would interfere with the molecular interactions between the resin-supported peptide and TFA. So in the future, we hope to successfully synthesize all 16 peptide biosensors that were designed and investigate to their fluorescent capacities with NEC4, and that all biosensors are to be purified using reverse phase HPLC and characterization provided using mass spectroscopy. And although the first one had a low yield for initially synthesized peptides, the new ones have seen an increase in yield in recent peptides. And with these tests, we will test the effectiveness of the peptides in an upcoming in vitro analysis to evaluate their interactions with NEC4 kinase. Finally, as a researcher, I would like to thank the Chemistry and Biochemistry Department of the Summer Undergraduate Research Program team at Queens College for its unwavering support and funding of research activities, and I would also like to extend my gratitude to Dr. Choi and Shao Ma for their generosity and their granting permission to use their mass spectrometer. And then these are my references and sources for the experiment. Thank you.